Hi everyone, welcome to my All for Science channel, and I'm back with this week's edition of Top Science News. The last time I did this, I had predominantly used slides and presentation, but this time I've decided to experiment a little and have put myself on the camera. But I'll also use slides in moderation. So the first topic that I want to talk about is regarding the Paris Agreement. And latest research shows that the Paris Agreement may be difficult to fulfill. The Paris Agreement of the year 2015 and signed in 2016, which had brought almost all the countries of the world on the same page regarding efforts to combat climate change, but later out of which the United States had opted to drop off, is now seeming like too ambitious a goal for the world. The Paris Agreement's long-term goal was to keep the increase in global average temperature to well below 2 degrees Celsius above pre-industrial levels, and to limit the increase to 1.5 degrees Celsius, since this would substantially reduce the risks and effects of climate change. According to a latest study done by a collaboration between the University of California and Carnegie Institution for Science, if existing fossil fuel burning infrastructure like power plants, boilers, furnaces and vehicles continued functioning as usual, they would emit enough carbon dioxide to exceed the 1.5 degrees Celsius climate stabilizing goal set out by the Paris Agreement. To make matters worse, there are infrastructures that have been commissioned and are going to be built in the future. And if these are taken into account, we might reach the 1.5 degrees Celsius threshold as early as 2033. The study has estimated that the existing infrastructure could release approximately 658 gigatons of carbon dioxide into the atmosphere. And out of those emissions, 41% would come from China, 9% from the United States, and 7% from the European Union. If emissions from planned, permitted, or under construction infrastructures are added, then the total would rise to 846 gigatons of carbon dioxide, which is more than enough to hit the 1.5 degree budget by 2033 and represents two-thirds of the 1,170 to 1,500 gigatons of carbon dioxide to reach 2 degrees of warming. The only way to halt this relentless surge of average global temperature is to retire existing power plants and factories way before their operational lifespan, develop technologies for large-scale negative emissions, and completely stop construction of commissioned infrastructures. So the second topic that I want to talk about is about something called the Great Atlantic Sargassum Bloom, and that its mystery may have finally been solved. The Great Atlantic Sargassum Bloom is perhaps the world's largest growth of seaweed in recorded history. It's made of a brown seaweed called sargassum, a kind of macroalgae, and forms a floating mat over the tropical Atlantic Ocean, stretching a whopping 8,850 kilometers from the west coast of Africa into the Gulf of Mexico. It's quite a recent phenomenon, first coming to the world's attention in 2011. And that is what's strange about it, since before 2011 there was not much sign of this algae except in patches around the mouth of the Amazon River in the summer and fall. Now, a team of scientists led by the USF College of Marine Science may have finally found the answer to the mystery. The blooms seem to thrive in nutrient-rich waters, with sea surface temperatures not too warm and just the right amount of salinity. Two sources of nutrients appear to be feeding the blooms. First is discharge from the Amazon River. This has been identified by looking at how much chlorophyll A was in the surface waters by using satellite instruments that scan the ocean in visible and infrared light wavelengths. Chlorophyll A reflects infrared wavelengths more than the darker water beneath and can thus be easily observed. In 2009, chlorophyll A levels spiked in the Amazon River plume, the part of the Atlantic Ocean that receives outflows from the Amazon, and they stayed elevated for most of the years that followed, possibly due to rising deforestation rates and fertilizer use upriver. Second source is upwelling along the coast of West Africa. Upwelling is when surface waters are pushed aside by strong prevailing winds, allowing deeper nutrient-rich waters to rise towards the surface. And this occurs there naturally due to the interaction of winds, ocean, and rotation of the earth. Sargassum is usually helpful as it provides shelter for turtles, fish, crabs, eels, and other marine species. But too much of the seaweed makes it hard for certain marine species to move and breathe, especially when the mats form near coastlines. When it dies and sinks into the ocean bottom at large quantities, it can smother corals and seagrasses. On the beach, rotten sargassum releases hydrogen sulfide gas and smells like rotten eggs, hampering tourism. Due to deforestation and heavy use of fertilizers not stopping anytime soon, there will be a constant influx of nutrients, and the researchers fear that such colossal blooms could be the new normal. So the next topic that I want to touch upon is regarding an expedition to study climate in which scientists will have to trap themselves in Arctic sea ice. This mission, called the Multidisciplinary Drifting Observatory for the Study of Arctic Climate, or in short MOSAIC, is a year-long mission to study the effect of climate change on the Arctic and how changing Arctic climate in turn affects the rest of the world. Scientists from 17 nations will participate in this mission while they go on a voyage to the planet's far north. But voyage sounds fun. This is not. The ship that is going to carry them is a German icebreaker called the RV Polarstern. For the mission, dozens of scientists will sail in a hulking ship packed with supplies and scientific equipment and venture right into the middle of the Arctic Ocean, anchor the ship to a large piece of sea ice and allow the water to freeze around them, effectively trapping themselves in the vast sheet of white that forms over the North Pole each winter. With freezing temperatures and shorter days, they will be racing against time to set up makeshift research camps on the ice, allowing themselves to conduct experiments which wouldn't be possible at other times of the year or by satellite sensing. By combining measurements on the ice with data collected from satellites, scientists hope to improve the increasingly sophisticated computer models for weather and climate predictions. The mission is being touted as similar to the International Space Station in terms of its unprecedented scale and ambition. The mission involves efforts from multiple countries and multiple disciplines like physics, chemistry, and biology. The expedition would cost $158 million and is being funded by US institutions such as the National Science Foundation, the Department of Energy, the National Oceanic and Atmospheric Administration, and NASA. The next topic is a good news since it gives us some hope, and it's regarding a new technique that has been discovered for permanent removal of HIV-1 virus. In a study published in the journal Nature Communications, researchers at the Lewis Katz School of Medicine at Temple University and the University of Nebraska Medical Center have reported that for the first time, replication-competent HIV-1 DNA, the virus which is responsible for AIDS, has been completely removed from the genomes of living animals. Current HIV treatment uses antiretroviral therapy, in short ART, to suppress HIV replication, but does not eliminate the virus from the body. So ART is not a cure and has to be administered lifelong so that HIV does not rebound, renew replication, and fuel the development of AIDS. The HIV lies dormant in the genomes of cells of the immune system, out of the reach of antiretroviral drugs, and waits for the right time to integrate its DNA into the genomes, at which point the cells get infected again and the disease reappears. The team of researchers have used a combo attack using CRISPR-Cas9 technology and laser ART to deal a double whammy on the virus. The first approach, that is CRISPR-Cas9 technology, uses a precise gene editing system to effectively excise large fragments of HIV DNA from the infected cells. The second technique, called a long-acting slow effective release ART, or in short the laser ART, targets viral sanctuaries and restricts HIV replication for extended periods of time. Both of these methods, when administered sequentially, have been able to get rid of the virus completely from the infected cells. This could be a groundbreaking discovery that could push us closer towards the development of a possible cure for human HIV infection. Okay, so the last topic for the day is again a good news since It's regarding a new gene that has been found linked to schizophrenia. A collaborative effort between Indian and Australian scientists has managed to hunt down a new gene associated with schizophrenia. The research, which has been ongoing for 18 years, has collected the DNA of thousands of people in India and found that those with schizophrenia were more likely to have a particular genetic variation. The study identified a gene called NAPRT1 that encodes an enzyme involved in vitamin B3 metabolism. When the NAPRT1 gene was removed in zebrafish, the brain of the fish could not develop properly and failed to divide symmetrically. And this finding is important since people suffering from schizophrenia also have defects in the corpus callosum, the part that connects the left and right sides of the brain. The team of Indian researchers was led by Professor Rangaswamy Thara, co-founder and director of the Chennai-based Schizophrenia Research Foundation. The research was published in JAMA Psychiatry and supported by Australian Government National Health and Medical Research Council NHMRC. This study provides us a new clue in the quest for causes of schizophrenia and for potential treatments. This brings me to the end of this video, and I hope you guys found something informative in this. If you liked it, do press the like button and subscribe to my channel. So until next time, take care.